Hi everyone, thank you for joining our webinar today. So my name is Dr. Poppy Nathan and I'm the Scientific Marketing Specialist here at Protein Tech and I'll be your host. And we're really lucky today to be joined by Dr. Mayank Sharma, who's a scientist at ETH in Zurich, who'll be telling us all about how to optimize immunoprecipitation of GFP-tagged proteins in plant cell extracts. But just before we come on to that, I'd just like to do a really quick introduction to Protein Tech in case you are unfamiliar with this. So Protein Tech is a leading life science reagent manufacturer of antibodies and immunoassays. We have an extensive portfolio of antibodies that encompass many different research areas. We also offer kits such as ELISA and IHC kits, as well as our cell separation kits and antibody labeling kits. So within our Protein Tech brand, we also have our humankind branded cytokines and growth factors. And all of our recombinant proteins are raised in HEC293 cells, so you can be confident that they'll have the native folding, post-translational modifications, and glycosylation that you would expect to see in human proteins. We also have our Chromotech branded nanobody-based reagents, which I'll come on to in just 1 minute. And lastly, we have a subsidiary of Protein Tech called Protein Tech Genomics that manufactures antibodies that are conjugated to oligonucleotides to allow for protein detection alongside your single-cell RNA sequencing. So all of our products are manufactured in-house, and we actually have the largest portfolio of antibodies for the human proteome, as well as a rapidly growing recombinant portfolio. We offer our antibodies in a 20 microliter trial size for convenience and testing. And we are ultimately a company for scientists by scientists. So as antibody experts, we have in-depth knowledge of immunoassay applications, and we can offer outstanding PhD level technical support to all of our customers. We also truly understand the importance of high quality validation. So all of our validation data is open access to our customers, and we try and perform our knockout and knockdown validation wherever possible. And finally, we have 7 inventory sites with manufacturing and research across 3 continents, allowing global next day delivery. And just because of the focus of our talk today, I just wanted to spend a couple minutes introducing our nanobody-based reagents. So nanobody products, or VHHs, are the antigen binding domains from heavy chain-only antibodies that are found as part of the natural immune response within alpacas. So they're only about 15 kilodaltons, meaning that they're actually about 10 times smaller than traditional antibodies, which allows for excellent tissue penetration. And they've also got extremely high affinity. So they have a much higher level of performance than conventional antibodies in applications such as immunoprecipitation, immunofluorescence, live cell imaging, and protein purification. So we've actually conjugated these nanobodies molecules that recognize fluorescent protein tags to agarose beads for use in IP, and we call these our nanotraps. So these are really beneficial for IP, especially for plant researchers, because unlike traditional antibodies, they don't have heavy and light chains that will show up in the gel, so you get much cleaner blots. And in general, recombinant proteins such as your GFP-tagged proteins can only be expressed to quite low levels in plants. Therefore, several grams of plant material are needed, which results in very large cell lysate volumes of up to about 10 mils. But due to the GFP-trap's very high affinity, the IP reaction can still be performed with as little as 25 microliters of slurry. Very highly concentrated plant cell extracts are also prone to bind non-specifically to all kind of affinity resins. However, because of the extraordinary stability of the GFP to the GFP nanobody complex, multiple washing steps can be performed and very harsh wash buffers can be used, supplemented with detergents or high salt levels. And the GFP fusion protein remains bound to the beads at all times, making sure that you don't see any non-specific background. And during the preparation of plant cell extract, the lysis buffer can also be supplemented with glycerol, reducing agents, and detergents to keep the GFP fusion protein in solution. But the GFP-trap tolerates even elevated levels of such harsh buffer ingredients without sacrificing its binding efficiency. And as I mentioned, because the expression levels of that GFP fusion protein in plant cells can be quite poor, the GFP fusion protein's concentration in the cell lysate may actually be very low, but sometimes, and which can sometimes be below the detection limit of even a Western blot. However, the GFP-trap can effectively bind to even the smallest amounts, such as lower than 1 nanogram of the GFP-tagged protein, which really enriches the GFP fusion protein to enable downstream Western blot detection. And then I've just got one slide with some data comparing how our GFP-trap performs to a conventional GFP antibody. And as you can see at the start, we've got the gel and the blots of the input fraction, the flow through, and the bound fragment. And as you can see here, the GFP antibody gel shows bands representing the heavy and light chains of the GFP antibodies, which you don't see in the blot in the gel from the trap. If you go down and look at the blots, then you can see that there is no protein left in the flow through of the trap, showing complete effective pull down, whilst the antibody still has a small amount remaining and it has a much smaller enrichment in the bound fragment than the GFP trap, showing that it has a less effective pull down. And all of this is because the traps have incredibly high affinity, as I mentioned, up to even 1 picomolar, which allows it to capture all of the GFP. We don't just have our nanotraps against GFP, we have them against a range of fluorescent protein tags, peptide tags, we even have them directly against some oncology proteins and more. And we are actually offering free samples of these to attendees of today's talk. So you can either scan the QR code there, or again, I'll have it at the end. And we can also send this out to everybody on slides within the next 5 working days. But now you've heard all about what a trap is, I can hand over to Mayank to show you how he uses it within his research. So Dr. Sharma completed his PhD at the Martin Luther University of Halle-Wittenberg. And in 2022, he received the ETH Zurich Career Seed Award. And he now works as a scientist in the Plant Biochemistry Research Group in Zurich. And his research focuses on unraveling the cell biology of starch granule initiation and biosynthesis in green tissues. So with that, I'll just say welcome. Thank you very much for being our speaker today. I'll stop sharing my screen and let you share yours and get on with your presentation.
Thanks, Poppy, for the nice introduction. It's a pleasure to be here and presenting this webinar. So let me share my screen. I hope everybody can see it. Okay. So far, so good. So hi, everyone. My name is Mayank, and as Poppy mentioned, I'm going to talk about immunoprecipitation of fluorescent protein-tagged proteins in plant cell extract and what and how we are doing this for a long time now. Talking about plant cells, they are complicated than animal cells, I should say. There are 2 extra subcellular compartments in plant cell, majorly chloroplast and vacuole, which make plant cells even more complicated, and a spatial compartment, which is cell wall, which makes plant cell rigid. And that's why it's not super easy to lyse plant cell compared to animal cells. The plant cells, they are interconnected and make tissue, and these tissue then later on form the organ of plants, or like leaves or roots or stems. A plant cell can contain several thousand different proteins. They are highly diverse in terms of their number and function and size and abundance. And purifying a protein out of these 1,000 proteins is tedious task. Why do we need to purify proteins? There could be several reasons. But one of or 3 major cause why one need a purified protein is either to understand protein function, or to determine protein-protein interaction, or for other practical applications like industrial uses or medicinal plant uses. But today I'm going to focus only on the first 2 aspects, understanding protein function or determining protein-protein interaction. And these are crucial for fundamental plant research, to defining molecular pathways and molecular mechanism, how these proteins are working together, if 2 proteins are interacting or not, what is the structure of protein. For all these, you need a purified protein from plants. So what was the classical approach? Like from early 80s, people were using this approach for purifying proteins. And this approach typically involves 4 crucial steps. First one is obviously to separate or to lyse the plant tissue by mechanical disruption or by enzymatic disruption of the cell wall, so that all the content of the cells, they are out. Then addition of buffers and protease inhibitors, because there are so many proteases in the vacuole. And then you precipitate and concentrate the proteins, which is still fine. And then more crucial step is the purification step, which needs sophisticated instruments like chromatography, centrifugation, dialysis, and electrophoresis. And in addition to this, it also needs specific antibodies sometimes. So if a protein is super low abundant in your plant cell, you will need an antibody against this protein. And then you can use this antibody as an affinity agent and then purify based on affinity purification. So this is the most time consuming step, I should say, can take days, and also requires sophisticated instruments like a chromatogram graph. Uh, but recent developments have simplified these processes. And then now we can actually purify protein in few simple steps. I will show you how. But coming back to antibody based purification, uh, we necessarily need not to have antibodies against the protein because they are alternatives, and alternatives are tags. So suppose if you want to purify a protein, and then if you can have a C-terminal, N-terminal tag on your protein, and these tags, they are versatile. There are antibodies available for these tags. They are small, so does not disturb the function of your protein too much. And then you can fetch out your proteins quite easily with the help of these tags. But these tags has to be genetically encoded and has to be expressed in the plant cells rather than in a recombinant systems like E. coli. Well, alternative to these small tags is fluorescent proteins. And I should say that so far there are more than 1,000 fluorescent proteins reported. The sequences of all these fluorescent proteins is available on FPBase, which is a database for fluorescent protein. You should definitely check it out to see how many variation of or variants of fluorescent proteins are available nowadays, and you can choose this one for your experiments. You should keep in mind while selecting these fluorescent proteins as tag for your protein that these fluorescent proteins should preferably monomeric. That means they should not form dimer or tetramer like GFP or RFP. So there are monomeric versions of these proteins now. They should be photostable, and always look for the origin of these fluorescent proteins. I have summarized these key aspects already in this chapter. You can have a look later on, but for now, why I'm focusing on fluorescent protein, because in addition to purify the proteins itself, it can serve another purpose, which is to determine the localization of your protein. So suppose if you just want to make one construct, and if you want to purify the protein as well as to see where it is localized in the plant cell, fluorescent protein tags offers a good alternative. So this is one example. I have fused frozen protein with one of my protein of interest, and this is how it is localized in the chloroplast in a plant cell. Nice, beautiful localization, and then we can use the same plant cells to purify this protein, to study it further, to test the protein interaction, and etc. Well, if we want to purify now these proteins, which are tagged with these small tags or frozen protein tags, we need specific antibodies. Unfortunately, the antibodies against these tags and also frozen proteins, they are easily available in the market from different companies. Um, his tags or his tag protein can also be purified with the help of these nickel columns, because histidine has a high affinity to nickel ions. Anti-flag antibodies are also available, and anti-GFP antibodies are also available. The good thing about anti-GFP and also other trap is that there are now nanobody combined with bead available, also with the protein tag. And these are called GFP-Trap beads for specifically for GFP and all the proteins which are derived from GFP. So works well with CFP or RFP. As Poppy has already mentioned, these anti-GFP or GFP-Trap beads, they have, instead of a full antibody, they have what we call nanobodies. These nanobodies, they just have the variable heavy chain component of the whole antibody, and they can bind to the antigen with a high affinity. And this is how hypothetical model of a bead with a anti-GFP nanobody looks like. And if you look these GFP trap beads under the microscope, this is how they looks like. So they are tiny magnetic particles which are actually combined with agarose, and then on the surface of these beads there are tiny nanobodies which can help fishing out your protein, which is quite fascinating to see that how these tiny particles can help you to fish out what you need the most for your experiment. Well, in principle, if we see a scheme of how this works, is that we have a plant which is expressing our protein of interest, preferably tagged with GFP or any other fluorescent protein, or YFP in this case. And then we fish out these proteins from plant cell lysate, and then can analyze with either with Western blot or with mass spectrometry. And I will go one by one how we address each step of the whole process and can fish out our proteins easily. So first step in this process is actually the expression of your protein of interest. So in this particular case, we need a protein which is tagged with GFP. So we need to make a recombinant expression. This can be done with protoplast transformation. So we isolate protoplast from the plant cell, then we transfect them with the construct which we make. And then these protoplasts, they express our protein of interest. Or we can use the infiltration of agrobacterium suspension, and this agrobacterium transfect actually infect the plant cells and then express the protein of interest. Or we can make stable transgenic lines with the help of floral dip or other suitable methods for specific plant species. This last method is time consuming, so generation of stable transgenic lines can actually take 6 months to a year, especially in Arabidopsis. If you are doing tissue culture, then it's a little bit faster. But transient transformation in Nicotiana is the most convenient and fastest way to express protein of interest and works well in our case. So if you'd like to have more or know more about these methods, you can have a look in these two publication or chapters. But these methods, they are available in the lab already in most of the labs. So you should be already familiar with this. So once we are done with the expression of protein of interest in the plant cell, the second step is extraction of this protein. So for extraction of this protein, we first need to select the tissue. So suppose if we are working with a protein which is expressed exclusively in the leaves, then we will obviously select the leaf tissue. If we are working on a protein which is expressed in root tissue, and if we want to find out which other proteins it is interacting with or want to determine its function, it's a little bit better if we isolate protein from roots rather than leaves. But if you don't have any options, or if the expression is super low in roots, then obviously we can choose leaves as an expression tissue. Well, once we determine that, okay, which tissue we want for isolation of our protein, then the second thing is obviously to select this tissue for homogenization. In case of Arabidopsis, if we are talking about leaves, we can either choose 2-week-old leaves, which works perfectly fine from these seedlings, or we can also choose a 5-week-old plant, which has large, big leaves, so it provides you good amount of material. But when selecting the material from these kind of plants, we need to be careful that we do not select petiole or any vascular tissue as such, because it contains a lot of proteases, which is not good for your protein extraction. And it is always advisable to combine young and old tissues together, so that if a protein has a specific partner in young leaf tissues, you can be, or you should be able to fish it out efficiently. If you are not using Arabidopsis, the second option is obviously using Nicotiana benthamiana, where we can express our protein of interest via infiltration. And in this case, I will recommend not older plants, but maximum 5 to 6 week old plant. This is an 8-leaf stage plant. And this is the plant which is perfect for expression of foreign proteins. If your protein is highly susceptible for degradation, do incubation of 48 hours. But if your protein is quite stable, then you can do incubation for 72 hours. But in any case, if your protein is expressed, you can quickly check with any fluorescence microscope which is available in your institute or in your lab. And if you see any fluorescence signals, that means your protein is more or less expressed. You can also do a Western blot to check if a protein of interest is actually expressed in these tissues or not. Well, once you identify tissue, you need to calculate the fresh weight, and then you need to homogenize the tissue in the extraction buffer. My recommendation for extraction buffer is 1 ml extraction buffer for 0.25 gram of plant tissue. This ratio works perfectly in our lab, and I think I always use this ratio for all the plant tissues, especially Arabidopsis and Nicotiana benthamiana. For homogenization, I recommend these two methods, or these two instruments, like tiny instruments, classical ones. Downs homogenizer, which looks like this. We have a pestle. You basically include your tissue in these kind of tubes, which are specialized homogenizer with a rough surface inside. And then you can crush the tissue with the help of your hand or specialized machines just look for a pestle binding rotator in your lab, and then should be fine. Or if you don't have this machine, because it can be, or these tubes, you can also use mortar and pestle, but in this case, you have to use liquid nitrogen just to make sure that by crushing um, the samples, you do not have too much temperature. And with liquid nitrogen, crushing plant tissue is much more easier. So once you have homogenization of your tissue, you can add extraction buffer, or you can already homogenize tissue in the extraction buffer, and then take out the lysate. Extraction buffer typically contains these components, so there should be a buffer. In this case, I use Tris HCl with a pH of 8.0, little bit of salt, a little bit of DDT, just to make sure that all the proteins or the non covalent interaction, they are gone, or they are not that stable. A little bit of detergent, Triton X100 in this case, it works well with Arabidopsis and Benthamiana. If you are using the solution or the beads for downstream application like mass spectrometry, I will recommend digitonin or DDM as alternative detergent, but never use SDS for homogenization if you want to do IP afterwards. I have heard many complaints from people who are using SDS-based buffers, and obviously SDS is a super strong detergent, and it will disrupt all the interaction in your lysate, and also the interaction between GFP and the nanobody. So no SDS, please. You can use any protease inhibitor, either commercially available tablets or PMSF as an alternative. But more or less, there's nothing more in the extraction buffer than these components. You can obviously play with the detergent if you are working with a strong integral membrane bind protein, but if you are not working with that, then it's fine. I think these ingredients of the extraction buffer works well. So after you extract, this is how the lysate of plant cell look like. It is completely liquid with this Towns homogenizer. Then you take this out, you centrifuge it at the maximum speed at 4 degrees. So make sure that after this step, even while doing homogenization, most of your tissue and the extraction buffer is on ice, or at least in the cold temperature. So once you take this out, you centrifuge it, you remove all the cellular debris as a pellet. Now also remember, collect a small volume of input so that you can compare this with the precipitated protein. um, so once you have done this, the next step is obviously the final or the main step of the whole process is combining the GFP-trap beads with the supernatant which you have just purified. My recommendation in this case is that for 0.25 gram of plant tissue, always use 25 microliter of beads, but this can vary. If you think that the protein which you want to purify is expressing super low in the tissue, then reduce the amount of beads, because then the protein will be more concentrated. And similarly, if you think that protein is expressing in abundance quantity, then you can increase amount of beads up to 30 microliter, but I will not go more than this with a 1 ml of lysate. Well, the next step after combining these two things is obviously the incubation, and this kind of rotating wheel is best at a 45 degrees angle. Um, this works best for 1 hour or 1.5 hours at 15 RPM. And incubation should be done also in the cold room if available. If it is not there, then it's a different thing. But if you have a cold room in your lab, in your institute, just put this thing in the cold room and do the incubation there. Um, this is because you don't want to have unnecessary reaction happening while you are incubating and then losing any of the interactions. Well, once you are done with this step, this is a kind of stand which you need to have in order to use these beads correctly. So these are especially made magnetic racks. You can buy them in market from different companies. So I will not say any particular name of the companies, but because it's just simple, tiny instrument. And this is how it works. Comes the magic part is that once you have beads and any protein solution or lysate or purified protein lysate, then you have to add this special magnetic strip to this rec. And now you can see already that all the magnetic particles or all the beads which were bound to the protein of your interest are now at one corner of the Eppendorf tube. And now if I will zoom in, you can see that these black things are actually your beads together with your protein of interest, hopefully. And you can also choose to have a centrifugation step to centrifuge these beads in one corner of tube. So like in the bottom video, and these beads now at one corner of Eppendorf tube. And once you are reaching this stage, I think 70% of your work is done for immunoprecipitation. Now you are safe. So once the beads they are interacting or associated with the magnet, you can easily take out the supernatant with the help of pipette and be confident the beads are not going to come out with the pipette. So you can do whatever you want to do with those beads and your protein now. So this is an example. I have these 6 different samples. Now I have beads with my protein. The next step is obviously to wash these beads again and again. So I will add wash buffer. Wash buffer is same as extraction buffer, but the concentration of detergent is half, and I choose NP40 as a detergent in case of wash buffer. So I wash this 2 times. So what we can do is this magnetic strip at the back, we can remove it so that the beads are relaxed. And then you add the wash buffer. Then again, add the strip so that beads are again associated with the magnet. And then you remove the buffer. You can do this 2 times with wash buffer or 2 to 3 times with this simple illusion buffer, which just has Tris and NaCl. Once you are done with washing, the last step is obviously protein elution. Now, protein elution is a relatively tricky, not tricky task, but if you want to have non-denatured protein, then it's a little bit tricky. But if you want to have denatured protein, then it's perfectly fine. You need to have SDS-based buffer. Lamelli buffer is perfectly fine. Boil the sample for 5 minutes at 95 degrees Celsius, and then your protein will be out in the solution. The alternative is if you want to have proteins under non-denaturing conditions, then you can use the antigen competition. In this case, you need to have anti-GFP nanobodies in your hand so that no, not nanobodies. You have to have GFP molecule or YFP or any fluorescent protein molecule in your hand. Add this to the precipitate. Then this molecule should ideally replace all the bait protein which you have, and you should have the protein of interest. But this is a tricky process. And with the fluorescent protein, I do not suggest this. This process works well with a small peptide tag like anti-flag or anti-V5, and you should use those kind of nanotraps if you want to do you want to purify in non-denaturing condition. But for STS page analysis, you need not to do that. This is how the beads looks like. If you look at the microscope in the fluorescent microscope, this is the protein of interest tagged with YFP, and these beads, they are perfectly shiny afterwards. And this is how the gel looks like. So this is just a simple Comasi stain. This is my input, multiple samples with a lot of protein from plant cells, and this is my purified protein, YFP tagged bait. And you can already see that out of these thousands of protein, purification of a single protein is already commendable. There might be a little bit of background, but this is something which you have to live with. You cannot get 100% purified protein. You can try to wash it more, but I think this is already highly purified version of tag protein. Now, to summarize what I have showed you is we had GFP-trap magnetic beads. We extract protein lysate from plant tissue. We incubate them together so that all the proteins and beads, they are together, incubated. Then we do the magnetic separation and then wash. So at the end, what we have is beads, GFP tagged bait, and any protein which is associated with our bait protein. So if I will simplify this in a simple gel, this is my input, 4 proteins. These are 2 proteins which were not binding to our proteins. And these are the proteins which are precipitated with the GFP trap beads, which is a GFP tag bait and my interacting partner. Now I can analyze this with Western blot, specific antibodies, or with mass spectrometry. This is one example where we have showed the interaction between two proteins. So in this case, VIFP was a bait. I have used a negative control CPFES-YFP, which is not interacting with my prey protein, and VIP flag, my prey. So I have used a different tag for my prey protein because if I will use the same tag, both proteins will bind to my GFP traps, and this is not ideal for the experiment. And this is an input gel where I can see the expression of my proteins which I want to express. So this is VIP flag in all the samples. These are my two bait proteins, via flex expressing via YFP expressing nicely. And this is my negative control CPYP FES YFP expressing nicely. So after immunoprecipitation, what do I have? I have via1 YFP, which was my bait, which is successfully precipitating the prey protein, which was VIP flag. The negative control was not able to fish out any of my prey protein, which kind of confirms that interaction between my bait and prey is kind of positive. One thing I would like to highlight here that you might experience some non-specific binding of your overexpressed bait or prey with the empty beads. But in this case, because I already used the negative control, this does not matter that much. But be careful if you are doing such experiments with empty beads. So this is a simple Western blot based analysis. We have detected interaction between these two proteins, but some people prefer to have mass spectrometry based analysis, where what you can do is you can send your beads to mass spectrometry facility in your institute, or if you are performing mass spec by yourself, you can perform on-beat digestion of all the proteins so that proteins are now digested in small fragments and do a shotgun proteomics. In that case, you will identify all the proteins which are coming out with the beads. But in this case, always remember to use a control. And control in this case could be a protein which is not related to the protein of your interest, but expressed in the same compartment. But these are some technical things which is for protein interaction analysis one has to consider. There is also a webinar on 28th of August by Simone Ziroli, and there you can learn about immunoprecipitation, how we combine this with the mass spec. So go check this out. But coming to the last part of the presentation, which is troubleshooting. So what you should do if you have problems with the methods. So one easy or one of the most common problem is that you don't detect any bait protein with the beads. There could be several reasons for this, but I have listed some of them. It could well be that your supernatant or the lysate, which you obtained after the homogenization of tissue, does not have your protein. And if your lysate do not have your protein, then you will not detect it in the precipitation. Make sure that you have used the correct tag. For example, GFP tags do not work with RFP tag, and RFP tags do not work with GFP tag very well. So make sure you remember the origin of your fluorescent protein, which family the protein is belonging to, and use a specific fluorescent protein trap to precipitate your protein. If there's nothing, always you should have a test control, which is working all the time, expressing nicely your protein, a soluble protein preferably. Check this. If this is not working, then there's some problem with the buffer. But if this is working, then it's a protein problem. Difficult plant samples are obviously difficult to IP the protein. Membrane proteins are also difficult, so you can try different detergent, different concentration of detergents. But these are the things which you have to figure out by yourself and trying different things. There could be too much background or unspecific proteins coming out with your beads. What you can do is you can just increase the number of washes. Number of washes with the simple elution buffer with the TRIS SCL and NaCl, and hopefully you will get rid of this unspecific background. And you should not worry about it because GFP-Trap and the bait interaction is super strong. And with these washes, it will not go away. So your bait protein will at least stay there. But if you are determining the interaction between two proteins, then it's a different thing. Then you should not have many washes because then the interaction between your bait and the prey will also be lost. The third thing is obviously no prey protein is precipitated with your beads. And I have people coming to me several times saying that my protein is not coming with my bait protein. What should I do? Well, the simple answer for this question is maybe there is no interaction between your protein and prey. So uh, assuming that you are working with two proteins and they are interacting directly and physically is not always true. So there is possibility that your protein interaction is not direct and not physical. Another thing may be that the prey protein is not expressed in high quantity, so that if you have 20 molecules of bait and you have one molecule of prey, obviously you will not detect it so nicely. To circumvent this problem, you can try expressing your protein or overexpressing your proteins in heterologous system. Nicotiana is a perfect system for plant proteins. You can also use yeast or E. coli cells. Or if this is also not working, then you may try different detergent. But I don't think this is needed if you are not detecting your protein coming out with the bait protein. Well, these are tiny troubleshooting, and I would like to show just two slides about what is happening with the GFP-Trap beads, how else you can utilize these beads. So I found this nice paper or nice essay, which is called Visible Immunoprecipitation Assay. So let's assume that BBS4 is one protein, which is your bait protein, and you are working with it, and you are using GFP-Trap bead to fish this out. And this protein is interacting with BBS18, and BBS18 is interacting with BBS8. And let's see now what happens here. So your protein, which is your bait, is nicely fished out with the GFP-Trap beads. And because this is interacting with this BBS18 protein, which is tagged with a different fluorescent protein tag, is also fishing out, you will see a nice ring of the second fluorescent signal as well. Because the second protein is interacting with the third protein, you will also see a nice ring of the third protein, which is now tagged with RFP. But if you are, if in the solution in lysate you don't have the middle part, like BBS18, you will not see signals from any of the protein, which is showing you a best example how you can use these GFP-Trap beads for imaging-based immunoprecipitation assay to determine the interaction between these two proteins. Well, this is one example how you can use the beads alternatively. Another thing which is more recent, this paper came out just earlier this year, and this was about cryo-EM. I know many people want to know the structure of their protein, and isolating and purification of protein is a tedious task. So what these people have done is that they have uh, made a long spacer, which is making a space between the bead and the GFP nanobody. And these spacer provide actually literal space which separates your protein particle from the beads. And what happened as a consequence is that in the cryo-EM analysis, the spacer and the magnetic particles are on one side, while your protein of interest is at the periphery of these spacer. And then you can actually use these particles, if your particles are homogeneous, for determining cryo-EM based structure. For example, here is a nucleosome structure um, analyzed by these method. I am not sure how good the resolution will be, but I'm sure that at least the basic structure should be figured out by this method. So this is one example how you can use these magnetic beads. It makes your life super easy. You don't need spatial instruments. Like the whole process from lysate to the purification takes just 2 to 3 hours. So give it a try and make your protein purification more simple. Yeah. So with this, these are the references if you would like to have or dig more deeper into any of these papers, you can take a screenshot. And with this, I would like to say thank you for your attention, and I am happy to have any question from you um, on any of these aspects.
Thank you so much. That was a really, really useful presentation. I'm not gonna lie, I myself am not a plant biologist. I come from an immunology background, so I feel like I've learned a lot myself as well. So if you would mind just to stop sharing your screen for me.
Okay, yeah, sure.
And then I can just run through some closing slides, and then we can come on to the Q&A, because we've had a really great discussion in the chat. I don't know if you've been able to see that, but lots
No,
of
I was.
questions in the Q&A box. I've tried to answer as many as I can, just on how the GFP traps work and anything to do product related, but there's lots about plant purification techniques that I'm sure people would love to hear your insights on. So I'll just share this really quickly. There we go. So I just wanted to take this time as well to introduce to you all a new product that